So I'm going to present on behalf of a team across the globe from Nigeria, France, and Canada. So the topic of the presentation is identification of QTL, controlling resistance to Anthracnose disease in water. So as you can see here, the Anthracnose is a very deadly disease for the yam production, especially in West Africa during around June, July, where the humidity is very high. So, but here you can see a kind of a resistant plant, and then here is where the disease is very high, and then can lead to 100% yield loss. And then the yam is in West Africa is considered as a cash crop, is also a food crop, and then a lot of people grow it across West Africa, also across the globe. And then we have a lot of species, more than 600 different species, but we focus mainly on 10, and then Alata is one of them. And then, as I was saying before, Anthracnose disease is one of the most important disease for Alata production. And then breeding through a classical is too long and too challenges. So here now we want to see if we can use if we can identify those QTL controlling those Anthracnose disease and try to develop some markers that can be used at early generation. So the way we proceed for the phenotypic observation is just from 1 to 5. So 1 is kind of like very resistant or tolerant a plant where we don't have any disease symptom. And then 2, around 25% of the plant is attacked. And then the 5 is 100% of the plant is attacked. So the material we use is 206 genotype, which were originated from biparental crossing. So we have 204 progeny and 2 parents, and then 1 is susceptible and 1 is resistant. So here we try to present the workflow, generally how we proceed for the QTL identification, the genetics map construction, and then so we have 1 female resistant, and then we cross it with the susceptible male. Then we have 204 progeny. We genotype them by using a GBS with our colleagues in France, Montpellier. And then we proceed for the SNP calling by using VCF Hunter combined with GATK. And when we proceed for SNP quality filtering, missing data, the read, the depth read. Then for the phenotypic estimation, we proceed for visual observation ranging from 1 to 4. Then we transform the data to quantitative by estimating the area under disease progression curve. Then we subject our VCF file that we obtain because Alata is, we are suspecting a ploidy level in the data. Then we subject the VCF file that we got to MapPoly. And then from there, we now proceed for the chi-square test performing by removing those markers that were highly distorted, and then we perform also some benphoronic tests. The final QTL detection was proceeded by using a SIM composite interval mapping. Then we proceed by using a QTL2. So here, the phenotypic observation for the data, we have a very good, very high irritability, around 0.71, but the coefficient of variation is very high also. And then by subjecting the VCA file to the MapPoly, we're able now to eliminate those markers that have high missing information. From 15,000 initial SNP marker, we got a 7,000, around 8,000. Then we exclude the 1,241 with high missing. The same method also was applied for the genotype, for the genotype, and then we remove a 25. We could have input the data, but we feel much better to remove those genotype with high missing information. Then we perform another analysis by estimating the benforoni, and then we excluded another set of markers that are very highly distorted, and then eliminated another marker that are kind of like redundant across the genome. And then we end up with a 3,000 around 3,000 SNP marker that are very good. Then we use it for the QTL detection. And then from there now we proceed for the pairwise recombination fraction to see if we have some marker that probably they are not well ordered from one linkage to another one. And then here we have a very good, before we are not having some very good pairwise recombination, and then we apply some algorithm to order those markers that were not really in the good linkage group. And then at the end, we got this very nice pairwise recombination that we use for future analysis. And then here is the table where we have the marker distribution across the chromosome. We have and then we have the total length per linkage group, and then the total distance. Here we have around 1,400, and then very similar to what close to the genome reference of ALATA, which is around 1,600. And then the average gap is the distance between SNP marker is very low, and then the maximum gap also across the linkage group is very low, not very high. And then we have a very nice genetics group. And then here you can see that the highest linkage, the highest length was observed on chromosome 2, and then we have some high density SNP marker on chromosome 4, which is also very similar to the genome reference, and then also with what some other colleagues has reported that chromosome 4 has the highest SNP number, and then the highest density chromosome SNP number. And then by applying the permutation through the composite interval marker, we're able now to identify four chromosomes, that 4 SNP markers distributed on 3 different chromosomes that are associated to YAM and Anthracnose disease. And then we have the position. Here I remove the composite interval because it seems a bit large, and then I put the gene function. So here we are currently focusing on these 2 genes because it has been reported in some other crop in potato, where this one they are playing a very good role in potato, especially in plant defense mechanism. So we are working towards validating this gene. And then you can see here now we now went back to the genetics map, and then refresh those markers that we identified to be associated with the YAM and Anthracnose disease. You can see those 2 on chromosome 7, one is here, another one is here in the same interval. The one on chromosome 15 is here, and then chromosome 18 is here in blue. Then we now check the interaction between the different chromosomes we have, just to see if they are unique, or some of those markers they are kind of like interfere in another one. And here, most of them they are kind of like very unique. And then the data were collected for 2 years, and then what we used to identify the QTL was a general blue. And then here now we want to see if the QTL we observe is kind of like stable from 1 year to another one. So we regress the QTL, the general QTL we observe by the year. And then we realize that most of the QTL, they're very stable, except one QTL obtained on chromosome 7. So currently we are not focusing on such chromosome because we can now use it to predict the YAM Anthracnose disease over year or over generation. Another activity we did again is we try now to look at those markers that we identify, how those markers can help us to predict the yield, they can help us to predict the disease resistance in the population we use. So here we can see that the presence of C is associated to susceptibility, because more the AUDPC value is high, more the disease pressure is very high. why the presence of T lower the disease pressure. So here now, we have already an idea about this marker that you have, how they can perform in the population, and then we are currently working with Intertech and EIB to validate this marker, and then we can deploy them into the breeding program for final using and then prediction at early generation. So I'm taking this opportunity to thank the polyploidy group to offer me this opportunity to share our works, and also thank our different collaborators and then the yam breeding unit. Thanks so much.